Outstanding. Thank you. Our folks are recording today. Good afternoon, everyone from College of the Canyons, specifically our outstanding Canyon Country campus. I'm really glad you're here for today's virtual Science Talk event. My name is Ryan Thule, and I have the great pleasure to work as VP of our College of the Canyons Canyon Country campus. And as the primary administrator for this vital site, I really enjoy the opportunity to work with many talented faculty, staff, and students. On behalf of our Chancellor, Dr. Van Hook, our Board of Trustees, and COC's hardworking faculty and staff, we at the college really enjoy nothing more than the opportunity to throw open the door to our campuses, to welcome in neighbors in our community, to visit and touch things hands-on and interact. But with COVID-19 plot twist surprises, this obviously isn't possible today, but we are nonetheless really excited to bring you what I think will be an intriguing and engaging and inspiring virtual event it's definitely thought-provoking and timely. And as we continue our series of science talks showcasing subject matter expertise, I think it's really fitting, and we'll keep that track record going. You're in for a special treat today. We have an amazing panel on COVID-19 from some preeminent faculty professors in COC's biology department. You'll hear more about them in just a moment. And I will turn it over to our Dean of Campus Services and Operations, our amazing Anthony Michaelides. He'll work as our MC and webinar coordinator today.
And here's some great stuff today. So I'm going to turn it over to the panel and take it away, Kelly.
Thank you so much, Dean Michaelides, as well as Dr. Ryan Thule and team from Canyon Country Science Center. I'm so proud and honored to be invited to speak to you guys today with my colleagues about this COVID pandemic, as well as the COVID-19 vaccine, which I think we're all pretty excited about. Today, we are speaking individually. However, it is one talk, so I'd like to give you just an overview of everything you're going to hear about today. First, I'm going to give an introduction to this virus, SARS-CoV2, and its associated disease. I will talk a little bit about natural immunity, which is how our immune system would normally respond when we are infected by a virus. And then I will turn it over to my colleague, Dr. Ramey, who will speak about vaccine development and specifically the COVID-19 vaccines that have been recently close to approval in the US and approved in Europe. And then finally, we'll finish with the all-important Professor Kelly Burke speaking about herd immunity. And we know that that's basically the key point we need to get to in order for us to be able to get back to campus and to get back to normal. So to begin with, I would like to discuss, wasn't moving. I would like to talk about the SARS-CoV2, which is the actual virus, and then COVID-19, which is in fact the disease caused by that virus. So it was not even a year ago when on December 31st, 2019, so basically New Year's Eve, when the Associated Press first reported a novel pneumonia-like disease happening in 27 patients in Wuhan, China. And I think that for most of us in the United States, we're like, hey, it's New Year's Eve, we're still gonna party. There's really nothing for us to be too worried about. And I don't think it made that much of an impression on us. But here we are, less than a year later, and you can see that we are now at a total of 63 million cases of COVID-19 that have been reported around the world, and that basically 2/3 of those are within just 10 countries, the United States being the leader, and I put that in quotes, in the world in the number of COVID cases. And I feel like probably this year, sadly enough, our New Year's celebration is going to have a very different feel to it than our New Year's celebration from the past. So just to speak a little bit about this virus, the SARS-CoV2 stands for Severe Acute Respiratory Syndrome Coronavirus. And I think we're all a little familiar. We've all heard that it causes breathing difficulties, coughing, and that's that respiratory syndrome aspect. It is an RNA-based virus. It's very tiny. If you were to take a piece of hair and look at a piece of hair, you can actually fit 1,000 of these viruses across the width of a piece of hair. And it's actually 10 genes are found within this virus. So it's pretty simple. Of those genes, and I'm gonna just draw your attention to the picture on the left, there are 4 genes that are probably the most important in the vaccine development. And I'm gonna focus on, and I believe Professor Ramey is gonna focus on the S protein. That's that spike protein, the little green one here, which decorates the outside surface of the SARS-CoV-2 virus. Now, when one of these viruses comes into your body, so we know that the transmission of this virus is either through breathing droplets or touching surfaces that are contaminated and then touching your eyes, touching your nose so that it's coming into the respiratory tract. This virus is looking for a docking station. It's looking for a location where it can attach onto a cell, enter that cell, and then it's going to basically take over that cell and hijack it to make more viruses. So scientists have discovered that the location of the docking station is something called an ACE2 receptor, which is shown here in this picture on the right. And for us to understand then, when this virus comes into our body and starts to replicate, what type of symptoms could be happening, we need to think about, well, where is that receptor or docking station found? Now, if I was to ask you and you were to guess, I think most of us, including me, would probably guess the lungs. And I was very surprised then to find out that in fact, this virus receptor is found on multiple organs throughout the body. And you can see that the lung is present about here, but that there's much higher concentrations of this virus that are found in the colon, the gallbladder, the heart, and the kidney. And this explains why when patients are infected with this virus, it is really not just the respiratory symptoms that we would associate with things like influenza, but we're going to see a lot more complicated side effects and complications happening in these patients. So in fact, doctors have discovered that in addition to the lung symptoms, that patients who are infected with this virus can also have heart problems, liver complications, kidney failure, attacks in the brain, which not only affect cognitive behavior, which is thinking and memory, but causing headaches, insomnia, and then of course the colon, which we saw had a very high expression. So the biology explains why this happens, but I think that this makes this disease a lot more treacherous than people first imagined. And one of the concerns that we have now is that when this virus affects those organs, could it lead to a permanent damage in these tissues and leave these patients with lifelong symptoms that they're dealing with? So this is another reason why having a vaccine against this is going to be so important. So now I'd like to transition into, okay, the virus got into our body, it attached to a cell, it made its way inside. How does the immune system fight viruses? And so our immune system, for those of you who are not biology students, which are probably many of you, consists of a lot of different organs, networks of tubes and cells. And the predominant cell are the white blood cells. And I've shown them here. There's actually more than 24. There's not a test later, so don't worry about it. Let's just ask ourselves, what does the immune system do? And it basically has three functions. The first function is to detect. So it needs to be out throughout the body 24 hours a day doing surveillance. So it's looking for things that should not be there. Anything that has gotten its way into the body, whether it's you ate it, you breathed it in, you got a splinter and it came in through your skin. After it detects and finds those foreign invaders, its next function is to destroy that. This is where the immune system ramps up and attacks and gets rid of the foreign invader in the body. And then lastly, and probably an overlooked function of the immune system, is that it's supposed to remember that particular foreign invader. And this is a really important aspect of the immune system because it protects us for years to possibly lifelong protection in the face of this memory that is formed after we have been faced with a virus or bacterial infection. So how do they do this detection? Let's talk about this. First, the immune system is surveilling, as we talked about, and they go up to cells and they actually, almost like Braille, they read the surface and they're looking for these things called antigens. And antigens are markers. The ones I'm going to focus on are ones that actually stick off the surface of the cells. And I think of them almost like little antennae. So they have different shapes, different sizes, and every different organism is going to have a different type of antigen found on its surface. So you guys can see in the picture here that this cell that came from a human cell has self-antigens that look sort of like little cylinders. And then if we look at this bacteria or this virus showing here, you can see that the shape of those antigens, those antennae, look different. And that is going to be recognized by our immune system as non-self. And as soon as our immune system says, oh no, there's something non-self, it knows it needs to attack and destroy that. So specifically talking about the COVID-19 virus, which is SARS-CoV2, we know that the S protein, remember I mentioned that before, that little green spike, that acts as a very strong antigen that our immune system can detect and react to. And in fact, the scientists working on this particular virus, this is a real life structure of what the virus S protein looks like, have found that the immune system responds very strongly to several different locations. So if you guys look closely, you'll see little red and pink markers on the S protein. Those are all the different locations that scientists have discovered the immune system responds to very strongly, and that makes them good candidates for building vaccines. So when we have detected, the immune system now jumps into action to get ready to destroy. But in order to do that, it needs to be able to alert the rest of the immune system in the body that, hey, I found a problem over here. So the cell that is originally detecting and finding these foreign invaders, I want you guys to cue in your mind the Jaws theme, dun, because they basically crawl up and they sneak up behind these bacteria and viruses. They grab them and then they pull them inside and they eat them. I guess eat, I'm going to put that in quotes because they're not technically eating, but they digest and they destroy them, which is a process known as phagocytosis, spelled for you guys here. This is some amazing scanning electron micrograph pictures showing white blood cells. It almost looks like they're fishing, right? You see their little arms, which are called pseudopods, reaching out, grabbing these organisms and bringing them inside. But the unique thing, so this is not just about digesting and getting rid of those. The unique thing that they do is after they have digested these parts, they take pieces of that virus or bacteria and they put them on their outside surface. And this is a thing called antigen presentation. And so you think of it as almost like displaying to the rest of the immune system, hey, look, this is what the bad guy looks like. It's almost acting like a wanted poster. Now you guys go find any other ones that are anywhere in the body and attack them. So I thought it would be great for us to go through an example of this, specifically looking at the process using the SARS-CoV2 virus. So everything begins with the virus infecting a cell. When it infects a cell, that cell is going to start to replicate copies and new copies of the virus leave. Now, one interesting thing that also happens at the point is this cell, and let's say that maybe this is a lung cell in your body, it recognizes that it is infected. And it recognizes that for the body as a whole, that's a bad thing. So it starts to take some of the viral pieces as the virus enters and displays them on its outside surface. And to me, this is almost like an SOS. It's sending a signal out to the immune system that says, hey, I'm not a virus, but I'm also a problem. You need to come deal with this because I've turned into a virus factory that's going to start producing more virus to attack the rest of the body. So we know now, once these new virus particles are being produced, that one of these immune cells, and I'm going to call this like a first responder immune cell, it comes in, it's going to grab these virus particles, pull them inside and digest them, just like we were talking about. And remember, what does it do after digestion? It takes pieces and it displays them out to the environment to show the rest of the immune system. So here comes in a cell that is saying, hey, I see what you're displaying. And now I'm going to use that information to tell all the other immune cells around me that this is the particular foreigner that we are looking for. So this is going to activate what I refer to as the second wave of the immune system. And man, when this thing gets turned on, you watch out, right? There's nothing more lethal in the world than the human adaptive immune system. And these are, rather than being something that just destroys randomly anything foreign, they are very targeted. They're like SEAL Team 6. They've been instructed specifically on the target to go find and destroy. And this includes sort of two different branches. The first thing that gets activated are things called killer T cells. The second thing that is activated are called B cells. There is not a test later, so don't worry about memorizing those. But I wanted to highlight that they do two different things. So this killer T cell is going to migrate over and attach onto that SOS signal. It has found a cell that is infected. It releases toxic chemicals, and it's going to kill that cell. This is a good thing. We want that cell to be killed because it is a virus factory, and it's going to just continue to produce viruses that are going to harm the rest of the body. But what are we going to do now about these free viruses that have already been produced? Well, activate that B cell. It's going to mature into a plasma cell that produces soluble proteins. These are called antibodies. Those antibodies are actually going to go and bind onto the surface of any free-floating virus, and that is going to neutralize that virus, meaning it can't infect other cells, and the immune system is going to come in and eliminate those. And just as a side note, that word antibody, you probably have heard that in the news. This is one of the molecules, and that's these little Y-shaped molecules. That's one of the molecules that we can detect for when we do a COVID test. So when you hear people saying, I did the antibody test, we're basically saying, does that patient express these proteins that match COVID virus? And if they do, that tells us they have been infected at some point in the past. Now, eventually, our immune system is going to eliminate the virus. This is the purpose of our immune system, and the immune system starts to shut down. We start to lose all of these antigen-presenting cells. I mean, they're not lost, but they're no longer active at the scene. And we have to go into our third function of the immune system, right? So detect is first, destroy is second. That third thing is to remember. And so we're now going to see the T cells and B cells starting to mature into cells that are called memory T cells and memory B cells. And the function of those cells is basically to remember COVID-19 virus, SARS-CoV2. So they are trained specifically to recognize just that one virus. And they can produce this memory that lasts in us for months to years. And in the case of some diseases like chickenpox, they can actually last for a lifetime. So this is something that we call immune memory. And this is something that is very important, that we want our immune systems to get to this, to be able to provide us that long-term protection. So my last thing that I'm going to talk about is how long does an immune response last when this happens? And so I want to, if you guys are not aware, which many people probably don't know this, but your immune system doesn't really become incredibly strong until the second time that you've seen either a bacteria, virus, or other foreign particle. So the first time you see it, which we call primary response, the virus infects, and then it takes the body actually a long time, 4 to 5 days, before it's able to start producing those antibodies. Remember, those are those little Y-shaped proteins that go and bind onto the virus. Also, in this graph right here, this IgM, IgG, those are antibodies. You can see that we don't produce that many. We produce a pretty low amount, and then sadly, they decline really quickly. So within 2 weeks, our immune response is done and over with, and we're basically not having protection. So this is in stark contrast to the second time our body sees either a bacteria or a virus, and this is called the secondary immune response. So that second time, you can see that the lag time to get started is 1 day compared to 4 to 5 days for the first time. Also, look at the amount of antibody we're making. It's a huge amount of antibody that is being produced, and it is declining very slowly. So this means that we are getting a very strong immune response that is giving us the ultimate protection against this virus. So just to look at them side by side, so you guys can compare the difference, you can see that where we want to get to with our immunity is always to the second response, where we're having a high amount of antibodies. And also, let me just overlay this with the amount of those memory cells, because remember, that's a really important function as well, is when we have that secondary response. Now, just to finish up, I'd like to say that the type of immunity I have been talking about is something that is called the natural immune response. So a natural immune response means that you were infected with the virus, you developed the symptoms of the virus, and potentially you developed the complications of organ damage, hospitalization, and death. There is another way for us to get to this immunity, to us to get to this immune memory and protection, and that is known as artificial immunity, which happens through vaccination. So through vaccination, you go into the doctor's office, you get a shot, sometimes two if you need a booster shot, and you experience brief symptoms of the immune system turning on, which can be fatigue, a little achiness, a mild fever, maybe lasting 2 to 3 days. And then you have the exact same immunity as if you had gotten the disease itself. So that double peak is going to be present with this as well. So of course, we would prefer to do vaccination for the safety because of these complications that happen with natural immunity. And just think about where we are right now. Almost 1.5 million people worldwide have died from COVID-19 or the associated complications of this disease. Right now, we're close to 64 million people who have been diagnosed with COVID-19, and this is being found in 180 countries around the world. So where we need to be and what we need desperately is a COVID-19 vaccine. And this is going to be so important for us in our ability to have a happy new year 2022, because by then, hopefully, if we have established the herd immunity that my colleagues are going to talk about, we can be back to normal and everything will go back to the way it was. Well, maybe not exactly, but close as possible to the way it was pre-COVID. So thank you for your attention. And I would like now to turn over my presentation to my colleague, Dr. Ramey.
Thank you, Dr. Cude, and thank you all for being here today. I'm Dr. Shane Ramey. I'm a biology professor at College of the Canyons, and I'll be teaching microbiology in the new science building at the Canyon Country campus when campus reopens. Just before the presentation today, I was reading that the World Health Organization announced that dogs cannot contract COVID-19, that dogs previously held in quarantine can now be released. So just to be clear, who let the dogs out? Just a little pre-pandemic humor to get my part of today's presentation started, but I actually have a story to tell. Some parts of it are sad, some parts of it are exciting. It does have a villain, it has heroes, but most importantly, it will have a happy ending. And the title of my story is Vaccinology, the race for a safe and effective vaccine for SARS-CoV2. Dr. Fauci recently said that the science of vaccinology, together with the tools of public health, are the things that will get us out of this. And this is that he's referring to is the COVID-19 pandemic. And as Dr. Cude just did an excellent job of explaining, without a vaccine, the toll that this pandemic is extracting on humanity is vast. If we look just at LA County alone, we lead the country by county in a number of cases. We have over 400,000 COVID-19 cases to date in LA County, and sadly over 7,700 deaths just in LA County alone. So this is certainly the sad part of my story. But with that, I need to introduce the villain, which of course is the SARS-CoV2 virus. This virus is the cause of the COVID-19 disease. And as Dr. Cude did a good job of explaining, one of the key components of this virus is its spike protein. And that spike protein interacts with that ACE2 receptor on many of our cells. And you can think of the ACE2 receptor as the lock and that spike protein as the key that gives those viruses access to our cells, causing those cells to create more viruses, which of course go out and infect other cells, causing damage and causing disease. Eventually, our immune system does mount a response to this virus, which results in the production of these neutralizing antibodies, as Dr. Cude was explaining. And these antibodies will prevent the viruses from infecting our cells. Before that happens, of course, we're susceptible to these pathogens, such as the SARS-CoV2 virus. If we're exposed to them, then that will cause disease, which will elicit a strong immune response and ultimately producing antibodies and these memory cells. So the antibodies will neutralize those viruses and help clear them from the body. And then those viruses, those antibodies will diminish in the body. So what's important in terms of immunity are those memory B and T cells that are produced. So this blue line here shows that these memory B and T cells stick around even after the antibodies go away. And that's the basis of the immunity that Dr. Cude was talking about, so that if we're exposed to the virus again, our bodies are able to mount a swift response, swift enough and strong enough to actually prevent disease and, of course, prevent death. What that will do, however, is create an even stronger immunity with even more of those memory B and T cells being produced, which will ultimately prevent disease. Now, that comes at a cost, and the cost, if we're exposed to the actual pathogen like the SARS-CoV2 virus, of course, is disease and often death. But we can achieve immunity without that through vaccination. This is what artificially acquired immunity does. It's immunity without disease and without death through vaccination. So we don't need to be exposed to these pathogens. We don't need to have exposure to the SARS-CoV2 virus to get immunity. We just need to get a vaccine, which won't cause disease, but will of course cause a strong immune response, which still results in the production of not only the antibodies, but of course those keys to immunity, those memory B and T cells. And with the second shot, many vaccines require 2 shots, and that's because the second exposure, in this case to the antigenic components of the vaccine, which I'll talk about, create even stronger immunity, even more of those memory B and T cells. If we look at the life cycle of the development of vaccines, it's a fairly long and laborious process. Vaccines to date have typically taken somewhere between 8 and 17 years to develop from and to be approved by the FDA for public use. If you look at some of these vaccines like the measles vaccine, hepatitis B, and others, we see that the time it took for those vaccines to be developed and approved was 10 years and longer in many cases. What we're seeing now with the development of the vaccine candidates for SARS-CoV-2 is that we're looking at some of those leading candidates being approved in less than a year, right? So let's look at some of those candidates and how they are achieving immunity and how they've been developed so quickly. And it's really all about that spike protein that Dr. Cude introduced earlier, that part of the virus that provides access to our cells. And if we can train our bodies just to produce those antibodies, which will of course neutralize those SARS-CoV-2 virus particles and prevent them from gaining access to our cells, then we prevent disease and of course prevent death. We don't need to use the whole virus in order to elicit an immune response and to train our immune system to recognize the virus. All we really need is that really good antigenic part of the virus, that spike protein, which will of course train the immune system. We can use to train the immune system so that the immune system produces those long-lived memory B and T cells and provide immunity. So here are, some of the good news, right? Some of this exciting parts of my story, and the heroes of my story really are these vaccines. And there are, the good news here too is that there are many vaccine candidates in development. In fact, and there are many different kinds of vaccines that are under development for this particular virus as well. I don't have time to go in to each of these different types of vaccines and vaccine candidates, so I'm going to focus on the 2 leading vaccine candidates for SARS-CoV-2 right now, which are the Pfizer-BioNTech vaccine candidate and the Moderna vaccine candidate. And these are both RNA-based vaccines. And this is also the part of the excitement about my story too, is that these RNA-based vaccines have the potential to revolutionize vaccine development, not just for this disease, but for many other diseases as well. And again, because they're RNA-based vaccines, there's no need for the entire virus, right, to make these vaccines. And so what's happened here is we've identified just a part of the virus that contains the information or the message for making that spike protein, right? And so that's part of the viral genome, right? So we've identified that component of the viral genome that contains that message, and we've used it to create mRNA or messenger RNA, right? So that messenger RNA just contains a little bit of information to make the spike protein and nothing more. These RNAs can be easily synthesized in a lab, and they need to be encapsulated in a tiny fat molecule called a lipid nanoparticle. And those particles stabilize the RNAs, and they also allow the RNA to be taken up by cells. So when someone gets an injection of the vaccine, those lipid nanoparticles that carry the RNA are able to make their way inside the cells, and then they're uncoated. Then the cell is able to actually use that information contained in the RNA to make the spike protein. Now, the spike proteins by themselves are harmless, so they can't cause disease, but what they will elicit is a strong immune response. And that strong immune response, again, will result in not only the production of antibodies that can neutralize the actual virus, but also those long-lived memory B and T cells, which establish immunity. So in this way, if someone who's been immunized comes into contact, isn't actually exposed to the virus, the body will be able to mount a very strong and swift response that will neutralize the virus, prevent infection, prevent disease, prevent death. So once immunity is achieved, there's no death and no disease as a result of the SARS-CoV2 virus. How has the development of these vaccines been accomplished in such a short period of time? Because we've been here, or we've gotten here in less than a year, and how do we get here so fast? And again, here's some more exciting parts of the story, really, because typically these phases of research and development and approval for vaccines has happened in a linear fashion. What we've done with this particular virus is we've stacked those different phases on top of themselves, and we're running these phases in parallel, right? And that's allowed us to really compress down the time frame for research and development for these vaccines. It still would be more than a year. So there's a lot of other components that have had to kind of come together for this process to work the way it has so quickly and efficiently, to shrink down those phases. And of course, on the top of the list is money, right? This is our modern moonshot, and that requires a lot of money. And the difference, though, with this particular moonshot is it's not a U.S. moonshot. It's a worldwide moonshot, right? This pandemic is not just affecting us in the U.S. It's affecting literally every country all over the world. So there's a lot of cooperation that's been going on that's allowed the vaccine development phases to move along quicker than normal. The RNA vaccines themselves don't require any virus. So growing these viruses in a lab is a laborious and time-consuming process. And so these RNA vaccines circumvent the need to do that. And research on these kinds of vaccines, fortunately, had been ongoing even prior to the pandemic. And that had allowed some key aspects of these vaccines to be figured out, like that lipid nanoparticle component of the vaccine that stabilizes the RNA and allows those RNAs to get into the cells. The genome for the SARS-CoV2 virus was sequenced very quickly, within about a month of those first cases being identified. and then that sequence was made publicly available immediately. And this allowed scientists around the world to start working on a variety of aspects of how to treat disease, how the virus causes disease, and work could start on vaccine development, right? And of course, that included identifying really good antigenic targets for these vaccines, which very quickly was determined to be that spike protein. So the gene for the spike protein was quickly identified, and the information to make these spike proteins actually has to be altered to make the protein stable without the virus. And that had been figured out in other coronaviruses, the SARS and MERS coronaviruses that had previously caused some epidemics. So that was fortunate as well. And those same alterations to the spike protein gene proved to be successful for the SARS-CoV2 spike protein gene as well, right? And then, as I already mentioned, these RNAs can be very quickly and easily synthesized in the lab without the need for for virus particles. And then, of course, last but not least, is the fact that the FDA is probably going to make an approve the emergency use authorization for multiple vaccines very quickly. So all of these things had to come together to make that vaccine development happen in such a short period of time. And so these vaccines are really the heroes of this story. They're going to be, and they're going to be what's the only thing that's going to bring this pandemic to an end. And if we look at some of these numbers, and Dr. Cude presented some of these numbers also, if we look at the reduction in the number of mortality, so the blue bars are deaths, and the orange bars are the projected reduction in deaths as a result of widespread use of a safe and effective vaccine, right? So we're looking at almost an eradication almost of deaths as a result of the COVID-19 disease. So we're looking at the end of the pandemic and millions of lives saved per year as a result of the widespread use of these safe and effective vaccines. So we're not at the happy ending yet, but I think the end of the pandemic is now in sight. So if we look at where we've come from, where we are now, and where we're going, going back to the beginning in December of 2019 is where we started to see those first cases in China of COVID-19. And about a month later, we had some in the United States. And that's also around the same time that the genome for the SARS-CoV2 virus was sequenced. By March of 2020, the World Health Organization had declared COVID-19 a pandemic. By April of 2020, we had over 1 million cases worldwide. By June, over 10 million. By August, over 20 million. And by September, over 30 million. During that time, though, vaccine development was ongoing, such that by November 20th, one of those vaccine candidates had already been used, or the producers of one of those vaccine candidates, the Pfizer-BioNTech vaccine candidate, had applied for emergency use authorization. In fact, the Moderna applied for their EUA just earlier this week for their vaccine candidate as well. So we have multiple vaccine candidates that have applied for their EUA. So what's going to happen next? Well, the an independent advisory committee is going to make a recommendation on December 10th to the FDA. This is an independent advisory committee composed of infectious disease and vaccine experts. They're not part of the FDA, but they're going to make a recommendation to the FDA. And I anticipate that the recommendation is going to be that the vaccine be approved, right? The FDA still has to make a decision. I predict that the FDA is going to make a very fast, very swift decision. I could be wrong about that, But my prediction is within days, the FDA will issue the EUA for the Pfizer vaccine. The Moderna vaccine is going to be about a week later. So assuming that the FDA also issues an EUA for the Moderna vaccine, it'll be about a week after that. So as soon as the first EUA is issued, those vaccines will be shipped out to the hubs across the country so that within a few days, immunizations will begin. So I predict that around December 14th or December 15th, we could actually start seeing shots in arms. If that happens, then about a month later, we'll start to see our first vaccine-based immunity in patients that get those, or individuals that get those first vaccine shots. I should also mention that both of the leading candidates, the Pfizer vaccine and the Moderna vaccine, require 2 shots. The Pfizer vaccine requires a shot, second shot, 21 days after the first shot. And the Moderna vaccine requires a second shot 28 days after the first shot. But around a month after the first shot is when those individuals will start to actually achieve vaccine or true vaccine-based immunity. And if all goes well from there, we could start to see the latter half of 2021 herd immunity achieved, and only then will we really be able to start to say that the pandemic is over. But that's the end of my part of the story, and the herd immunity part of the story is going to be discussed by Professor Burke. So again, thank you all, and I will now leave it to Professor Burke.
Hi, everybody. Thank you. I'm happy to be here too, and just kind of round up the end of our talk here and the importance of achieving herd immunity in our population. So let's take a quick look at some of the diseases that have been around for a really long time, or were around for a really long time, and were really important. None of these diseases were ever diminished or squashed or eliminated simply through what some people are calling natural herd immunity. So I'm going to talk a little bit about the myth of natural herd immunity versus what actually herd immunity is. So smallpox is was a serious disease. There were probably, hundreds of thousands of cases every year. In 18th century Europe, they predict there are about 400,000 cases per year. In the 1950s, vaccines were developed, and this was eradicated by 1980. It's the only disease that has ever been eradicated, and it was only done by vaccination. Some of you may know about polio. My mother had polio. She had a very mild case of polio, but I remember hearing stories about polio and how they would get off school for the summer and go back in the fall, and their classmates were missing because they had passed away from polio over the summer, or they were in iron lungs. So you can see the picture here of the iron lung patients. There are still patients who are actually living in iron lungs with assistance today. Polio was eliminated in the US in 1979. The vaccination was developed in the 1950s. And it's almost been eradicated. There are still some pockets in Pakistan and Afghanistan, as you can see by the red arrow. This is where the efforts are being made now to vaccinate and make sure that we eradicate this disease as well. Measles is an interesting one. In the early, up until the 1950s, again, 1960s, when the vaccination was made, almost every child in the world got the measles. It can be deadly, but it also, sort of more importantly, has a lot of secondary effects that are very dangerous. Through vaccination, it was pretty much eliminated in the United States by 2000. But we have outbreaks of this, and we have outbreaks of the measles because travelers bring it from other areas, and there are often pockets of unvaccinated populations where the vaccine rates go down. And so the disease emerges. And we'll talk about how that can happen in a population. In 2019, in the United States, we had a few outbreaks, and these were in those populations where vaccination was decreased. So the myth of natural herd immunity, you may have heard about this, and there are countries that have tried this. So that's the idea that you're just going to let the disease run its course. Eventually, everybody will become immune, and then everything will be fine. But we've never had a disease ever that has been conquered like this. In reference to letting it take its course, you can see a couple of quotes that really sort of point out this myth that there is nothing that natural herd immunity really accomplishes. And in that timeline of trying to get to natural herd immunity, there are a lot of deaths, a lot of hospitalizations, and this can really have a big impact on the public health systems. Sweden was a country that tried to do natural herd immunity through this COVID pandemic. That was not successful. Their first wave of the virus did not prevent a large second wave. And currently they are very much doing a lot of the same public health measures that other countries have already done in order to get this a little bit more under control than it has been. So what exactly is herd immunity technically? Well, herd immunity by the World Health Organization and by most public health experts, this is immunity that's acquired through vaccination, and it protects the entire population as long as there is a certain threshold of vaccination. So you need to have a certain threshold of vaccination in order to achieve herd immunity. Sometimes this is being called population immunity now, and it's even being called community immunity, I think in an effort to get people to take a little bit more ownership of this and think about their own communities and protecting their own communities. So what happens with vaccination and herd immunity? If no one gets vaccinated, and you can see the red dots are the sick people, and there's no vaccinated people in that little image. If no one gets vaccinated, the disease is going to spread easily. So it's going to go from the sick person to the susceptible person, from that susceptible person to another person, until a large population of the people are getting ill. And that is what is happening right now with COVID-19 and what happened in smallpox and the measles and all of those other types of diseases. Then when some people get vaccinated, the disease will spread a little bit slower. So if you have a population where some of the people are getting vaccinated, that disease may spread a little bit more slowly, but it doesn't get eliminated. And eventually people are going to get ill regardless. Then when most people get vaccinated, what happens is that you are protecting susceptible populations. So people who are immune are not coming in contact with the virus very much, and people who are sick are not coming in contact with people who are susceptible. So that herd immunity protects the whole population. Another little graphic kind of shows it. You can see that on the left, if only some get vaccinated, that red person in the middle, eventually a lot of people are going to get sick. But if most get vaccinated, that spreading is contained. So the idea of vaccinations really is to break the disease cycle and to break the transmission of disease to susceptible people. It's also a social agreement. And this is really sort of an important consideration that we've had to think about in a lot of ways when we've talked about these public health measures. Why do we wear masks? Why do we social distance? Vaccinations are always, and those kinds of things are always to protect yourself, but in a greater sort of moral and ethical way, they're to protect the vulnerable populations around you. So if you think about the people in your life, in your community, you probably know people who have newborns that cannot be vaccinated. You probably have elderly people that may have all kinds of secondary diseases that may not allow them to be vaccinated. You can have cancer patients. You know, people in your family and your friends who have cancers, they may not be able to be vaccinated. Transplant patients, people that are immunocompromised, so people that have autoimmune diseases, people that are undergoing immune therapy or cancer therapy or other chemotherapy. And then there are also people that are just born with genetic deficiencies and don't produce a good immune response at all. So when we vaccinate and we try to achieve these herd thresholds, we're really trying also to protect those vulnerable populations. So it's for our own individual selves, but it's also for our communities. So you probably heard about R naught. This is the R subscript 0 or O. And then sort of its friend RT, or sometimes it's RE. Well, the R naught is the transmission rate. It's sort of the reproductive rate of the virus. So how many people are going to get sick, right? If I'm sick and I'm out there, how many people might I infect? So it's measured in a completely susceptible population, which isn't always sort of what is true, which is where the RT comes in, and I'll talk about that in a second. But if the R naught is less than 1, the disease will stop spreading. If the R naught is equal to 1, the disease is sort of endemic. It's sort of static. There's a certain level in the population, but it's not really increasing. When the R naught goes to greater than 1, then the disease is spreading and outbreaks are starting to occur. So a little graphic here, if the R naught is 2, and I'm the sick person, I'm going to give that to two more people. If the R naught is 10 and I'm the sick person, I'm going to actually spread that to 10 different people. And the R naught is very specific to different diseases, though they all have different sort of R naughts. And this can show you that. So in this graph, if you look in the center, here's some of the diseases that I've talked about. So the R naught for measles is very high. It's 12 to 18. So if I have the measles, I'm going to easily give that to 12 to 18 people. Polio, 5 to 7, smallpox, 5 to 7. And the regular run-of-the-mill flu is about 2. For COVID, the measurement right now is between 1.5 and 3.5. But this is a little bit difficult to tell because there are a lot of factors that come into play when figuring that out. And we don't have historical data. So we have a lot of historical data on the measles, on polio, on smallpox, and even the flu to really hone in on what the R naught might be. So it's a modeling statistic, but it isn't always easy to come to. Now, if we look at herd immunity, how is this related? Herd immunity is the threshold of vaccinations needed to stop transmission. So because measles is highly infective, if you look at the little formula there, R naught minus 1 over R naught, that's how the herd immunity, the threshold is calculated. You need a very high threshold in the population. So 95% of the population has to be vaccinated to help stop the measles. And this is why in some communities that are under-vaxxed, they have outbreaks. For polio, about 80%, and the same for smallpox. For influenza, it's a little bit interesting because the R naught is 2, which means about a 50% calculated herd immunity. But because the vaccine that we have is not as effective, it's only 40 to 60% effective. In reality, you need more people to be vaccinated because they're going to come across susceptible people more often. COVID-19 right now, they're thinking it's about between 60 and 70% vaccination rates. But when we look at COVID-19 and herd immunity, we don't really know all the answers yet because it's very complicated. The R naught and the immunity have a lot of uncertainty because this is a new disease, right? It's an emerging disease. We don't know the virus properties completely. We don't know the pathogenesis of the virus, or even in some cases, transmission, right? We're still learning about all of these things. Then as you're measuring the R naught, the susceptibility changes over time because we have asymptomatic and mild cases. We don't know what those numbers are. We're learning about those numbers, but those people could be spreading the disease. People get sick and recover, so that changes the immunity in the population. It does increase it to some degree. How much, how long, we don't know all the answers to that. And now we're instituting disease control measures, and we did this right away in many places. So masks, travel restrictions, lockdowns, our social distancing, all of those things helped with disease transmission. Therefore, to figure out a true R naught is really difficult, and more data is needed. Now, RT, on the other hand, is sort of the real-time current reproductive rate of the virus at any point in time. And so scientists and public health officials, vaccinologists, they're all looking at the RT in real time. So the graph on the left shows the United States and what the RT was in March. And the orange is sort of between 1.5 and 2. The pale yellows are between 1 and 2. So you can see at the beginning of the pandemic, California and many places in the United States were already over 2. By July, we had reduced down to around 1. And so that is a very good rate where it's not really increasing as much in the population and not being as transmitted as much. And now in November, it's slightly increasing again. So scientists have to take this data and look at this data over time to try to figure out what is truly happening as far as transmission. How do we calculate the vaccine rates and how many people need to be vaccinated, et cetera. So it's a very dynamic situation. It's a situation where people are learning more all the time. So the way forward, if we're going to try to achieve herd immunity, is really thinking about what we continue to do and what will happen in the future. So as Kelly said in her slide, you know, hopefully a year from now, we're going to have those classrooms filled again. We're going to have our events and we're going to be able to get to some normalcy. I don't think it will ever be quite the same as it is now. And I think having this pandemic will only sort of let us know that this is possible in the future as well. So we have to continue wearing masks and doing our hygiene and social distancing because the rollout for the vaccines will happen incrementally. Not everybody will have that immunity right away. People won't know if you've had the vaccine. So again, those are those sort of social agreements. We still will have susceptible populations that will need to be protected. So we cannot stop those public health measures. In addition, the vaccination is coming out. So when you think of the United States alone, there's about 331 million people. And if we need about a 60 to 70% herd immunity threshold to stop the disease and to decrease transmission, that's about 231 vaccines. So that's a lot. There's, you know, Shane talked about the timeline and all the new vaccines coming out. It's hard to find really good information about what that's going to look like. So I've seen things like, 150 million doses by the end of spring. I've seen 70 million doses a month. But remember, you also need 2 doses. So 150 million doses really only covers 75 million people. So there's a lot of work to be done, a lot of cooperation to be done. In addition, looking at this long term and looking at future pandemics, we need to make sure that additional vaccines are created and that the availability is global, not just in, you know, countries that can afford this. We obviously need to increase better medications and therapies like using antibody therapy, increase testing capacity for both antigen and antibody testing to test for immunity, and increase contact tracing. Countries who have done this well, this increased testing and increased contact tracing, have really shut down this pandemic. The United States has had some trouble with this, but countries like Cambodia, Thailand, Australia, New Zealand, China, Japan, they've done an excellent job with the testing capacity and contact tracing. And this has really put them in such a good position for getting rid of this disease. And finally, we really have to think about this as a global situation and cooperating with other countries. The healthcare infrastructure is great in many countries and not so great in other countries. And those countries where that infrastructure is not there are always going to suffer. And the people there are always going to suffer, and they're always going to be a source of disease for other nations. So if you want to be selfish about it, you can think, well, you know, this is where a lot of other diseases come from because they don't have the same controls. But the real thing to look at is how do we help other countries achieve the same kinds of levels of immunity, herd immunity, vaccine, contact tracing. So it really does need to be something that we all cooperate in and that we're all willing to do. And it's a social agreement, not just with our own communities, but the world at large. So I'm very excited about what will happen in the future. And herd immunity and our immunizations really are the way out. So we'd like to thank you all, Ryan, Anthony, Jamie, Sean, and actually Maria and Nick, who are behind the scenes. And of course, all the first responders out there, if you're in the audience, we thank you so much. We always want to pay homage to Dr. Fauci and all of you for attending today. Thank you so much.
Great. I think we're going to go right into Q&A. Looking at the time right now, we have probably maybe 20 minutes or so. I'm looking at, I've been following the Q&A. Those of you who have Q&A open, lots of the questions have been answered. Our panelists have been answering it behind the scenes, so you can look in there and see the questions and answers. I'm going to read a few off that came in and throw them to the panel and see if we get some answers. Some of them have been answered again, but you know, they may just want to give the summary version of it. So if someone had COVID-19 previously, should they still be vaccinated?
I can take that question. So the short answer is yes. There's no downside to getting the vaccine, even if you've previously been exposed to the virus, even if you have had or may have had COVID-19 in the past. There's no downside to getting the vaccine. And as I also pointed out, and getting the second shot or exposed the second time will create even stronger immunity. So there's no downside. The upside is you'll have stronger immunity as a result.
Great. Okay. Does it matter which vaccine you receive? They've said several will be coming out. Also, let's see, can they have possible negative side effects if you get two different ones?
I can speak to that. I think that when you get the first vaccine, you're going to get the same one for the second vaccine. I don't think that they're going to be swapping out vaccines, and I don't think that, you know, that might not matter so much, but that's normally how it's done. You would get the same vaccine. Side effects are going to be mild. Well, they've talked about them being a little bit more extreme. I don't know that the different vaccines have different side effects.
Okay. Can someone who has been vaccinated still infect others who have not been vaccinated?
Okay, I'll take this one. If you've been vaccinated, then your body will not harbor an active infection. You will no longer be someone who's carrying a high viral load that would be necessary for you to spread this to somebody else. So that is one of the main things of being vaccinated, is that not only protects you, but it's going to help us to create that herd immunity and to protect people around you.
Okay, what are the panel, expert opinions on travel during the holiday season? Is there a safe way to travel currently, or is it better to postpone trips at the moment? Sorry, that's a tough one.
No, it's not. For me, it's not tough. My personal opinion is that over the next several weeks and through the holidays, everybody needs to just sort of stay home and just do the best they can. I think that, you know, if you absolutely have to travel, then wearing masks and taking hand sanitizer and doing all of those social distancing and practices that you need to do if you must travel. If it's optional travel, it really is in the best interest of yourself and in public health to not do that at this time. It's very hard, but it really is. Hopefully, a few more weeks of this, and then we'll start seeing the light at the end of the tunnel after the new year.
Great. If a person has had COVID or been exposed to someone in the same household with COVID, is the vaccine still recommended?
Yeah, this is a similar question, I think, that I answered, and the answer is yes. I think at this point, again, there's no downside to getting vaccinated, even if you've already been exposed to the virus previously.
Okay, I am reading, let's see here, it's a little bit longer. Do you believe that reducing stress through some type of medication or relaxation exercise, and perhaps not eating processed foods, sugars, drinking alcohol, smoking, and so on, would strengthen a person's immune system in terms of COVID, obviously?
I can go, just because this is a topic that I talk about in my cancer class as well. These are all good, healthy habits. The stronger your body is overall, the healthier you are, the better your immune system is functioning. So eating healthy, having antioxidants in your diet are all good things, but nothing has been done to show that that prevents you from getting the SARS-CoV-2 virus. So these are also, these are all great precautionary things. I don't think there's any harm in doing them. I think it's always great to be as healthy as you possibly can, but there's no guarantee that that is going to protect you from getting the coronavirus.
A question about masks. Are silver fiber masks better than regular cloth masks? Or for that matter, paper masks?
I can answer this, I guess, as the resident sewer in the group. There has actually been a lot of evidence that has talked about different fabrics. And there's kind of like two different things that they look for. One is like the breathability, because if you can't breathe well through it, then people aren't going to wear them. People are going to like pull them down under their nose, or they're not going to be able to wear them for long periods of time. And then you have to match that against the pore size and its ability to be able to block droplets or to block the access of the virus. And so right now, what they're saying from all the studies I've seen that are the best are basically a tight knit cotton. Those are the ones that have enough breathability, but also have the pore size small enough to effectively block droplets. But again, remember, even when wearing your mask, you want to maintain that 6 feet of social distance, because it's not like that mask is 100%. It's not like you have a piece of wood in front of your face that nothing can go through.
I'll just add to that just a small amount. It protects you with the droplets, because then the droplets dry and then the virus can't survive. Silver tends to be antibacterial. I'm not sure about antiviral because of the mechanism of it, but that's sort of a different story. I don't know that silver is going to help any more than just the cloth.
Ooh, I actually have one other thought. You can also get masks that have a filter insert. And those filters, which they're called like P2.5, which tells you that they take out particles down to 2.5 microns, are actually also very effective in helping to block the passage of particles back and forth.
Okay, how long will immunity from the COVID vaccine last? And I think I saw that somewhere answered, but a quick answer on that one.
So I was sorry. I don't know that there's an answer to that yet. Truly, there's some conjecture, but I think the honest answer is we don't know yet, and time will tell. So the vaccines that are vaccine candidates that are going to be approved, probably be approved very soon. A year from now, we may start getting some of the data that will indicate whether or not maybe annual vaccines will be necessary for this particular virus. If we look at the kind of virus that it is, it's a coronavirus. It's different than the types of viruses that cause influenza. So, you know, annual influenza shots are recommended because there's differences in those flu strains from year to year as a result of a couple of different things, but as a result of changes in the mutations in the genome of the virus, as well as the type of immunity that is achieved from the vaccines. And we're not sure about this particular coronavirus. Other coronaviruses don't tend to mutate like as quickly as influenza viruses do. And I have read some information that have been several mutations for the SARS-CoV2 virus have been identified over the past year or so. However, none of those mutations have altered that spike protein that I spent a lot of time talking about, and Dr. Cude spent a lot of time talking about as well. So although there's mutations that have been identified, they don't, so far we haven't identified anything that would cause immunity that's achieved through vaccination that use that spike protein as an antigenic target would wane over time, but time will tell.
Excellent. This is a good one regarding the actual vaccine itself. Why some vaccines need to be stored in very low temperature?
I'm going to take that one also.
I see that one going.
So not all the vaccines candidates that are being developed require very low temperatures. If we look at the two that I focused on, the Pfizer-BioNTech vaccine candidate and the Moderna vaccine candidate, those both being the RNA-based vaccines, the Pfizer-BioNTech vaccine requires storage at -70 degrees Celsius, which is super cold. That's essentially dry ice kinds of temperatures. And but the Moderna vaccine candidate can be stored at -20 Celsius, which is your freezer in your kitchen temperature, basically, very close to 0 degrees Fahrenheit. And so not all of these vaccines are going to require super cold temperatures. The reason that these RNA-based vaccines require low temperatures is because of that lipid nanoparticle that I talked about, and the stability of the RNA molecules that the vaccines are based on. So the information on those lipid nanoparticles is actually proprietary. So Moderna has the lipid nanoparticles that they have created are actually more stable at warmer temperatures, right? So they don't have to keep them as cold as the Pfizer vaccine candidate does, because there are some differences in the type of lipid nanoparticle that they're using to encapsulate those RNAs in. And that's likely to change eventually too, as Pfizer will probably figure out a better formulation for their lipid nanoparticle eventually. So it's possible that a future version of their vaccine won't require such super cold temperatures either.
Great. Here's a technical one that I can't answer myself. How can I best explain to others that these mRNA vaccines do not alter our DNA? Kelly.
Okay, I guess as the resident geneticist, I'll try to tackle this. So mRNA is a particle that is used to make proteins. It's basically an instruction sheet. And when this mRNA is entering the cells as part of the vaccine, it's basically going to go into the cell, be read as instructions by the cell, and then it will be turned into protein to make that S spike that is going to go out and alert the immune system. But then what cells do with mRNA is they destroy it. They get rid of it because it is a temporary copy. So they're not going to keep this around. They're not going to store it down inside the nucleus in the DNA. So it doesn't have any ability to actually become integrated into our chromosomes and our genes and to affect our DNA as humans.
Great. Are children able to get COVID-19?
Yeah, everybody can get COVID-19. We have seen children with COVID-19. They seem not to get it as much or as seriously. There have been some very serious side effects of COVID-19 in children that are associated with COVID-19. again, the pathogenesis of this virus is not completely understood. So why some people get it and other people don't, why children don't get it as much. Somebody was asking about asymptomatic people, why they are asymptomatic and don't get it, but they can still transmit it. These are things that we don't know about the virus 100% yet. And time will only tell, right, when we learn more about the pathogenesis and also how the vaccine is working.
And I could add just one potential mechanism that they're working on for why children are not as affected as adults, but it's, again, very preliminary. If you guys remember from my presentation, I talked about the ACE2 receptor, which is that docking station for the virus as it comes in. And they found that that particular docking station receptor doesn't seem to be as expressed in children yet. So they haven't turned that on. It isn't as high levels in their different organs. And that might be why they are a little bit more protected than adults, which have very high levels of that docking station. But again, preliminary data, so not fully understood yet.
Okay, gosh, we have a few minutes left. I'm going to try to get to at least a few more. We'll see. Hand sanitizer kills off good germs on our skin. Could this cause problems in the future?
I'll take that one. Yes, it depends on the hand sanitizer that you're using. So if you're using something that is ethanol or isopropyl based, we haven't seen a lot of resistance increasing to other microorganisms. When you're using other types of chemicals, there has been some increase in resistance. So a chemical that was out in a lot of hand sanitizers a while ago was triclosan. And that was associated with increased resistance to antibiotics. So there is potential for that, but for the most part, things like bleach, although don't use that on your hands, and the ethanol, there has not been quite the same amount of association for resistant organisms. And we're really talking about resistant organisms, sort of bacterial resistance to antibiotics.
Okay, is it possible for an individual to be immune to the virus naturally? As a young, healthy, athletic male with asthma, I've been around individuals who have had COVID, including family members, and have not gotten sick.
Okay, I'm going to try to think about this logically as a geneticist. And I can tell you that there might be more than one risk factor associated with the acquiring of the infection and getting sick that would make certain individuals more susceptible than other individuals. We see this with lots of things. I can give you an example. Lung cancer, since I'm a cancer specialist. Not everybody who smokes gets lung cancer, even though we know that smoking is 90% of the cause of lung cancer. So what is the difference? There's some genetic difference that is making those individuals more susceptible or less susceptible. And there's probably a similar mechanism working with the coronavirus that we don't understand yet, which is making some individuals more susceptible and other individuals less susceptible. And then there can, as you hinted at, asthma, and we know diabetes and other sort of underlying conditions can heighten your risk to getting these, but there are probably other factors aside from just having these underlying conditions that are playing a part that we don't really understand yet.
Several questions, I'm going to combine them all into one, asking about the side effects or possible side effects of the vaccine. Is it worth the trade-off to get the vaccine versus possible side effects and things like that? Obviously, you know, the question will continue as we get into the vaccine being distributed as well.
Yeah, I think, and Professor Burke talked a little bit about this earlier too. There are going, in many people who get the vaccine, there are going to be some side effects. I want to emphasize though that you cannot get the disease, you cannot get the COVID-19 disease from the vaccine, right? You can get side effects that might make you feel like you're getting sick or a little bit sick after you get the vaccine. And that feeling is a result of your immune system being kicked into overdrive. And that's exactly what you want your immune system to do when you get a vaccination. You want your immune system, as Dr. Cude discussed, to produce those antibodies and as I also talked about, to produce those memory B and T cells. So the feeling of feeling kind of sick, some aches and pains, maybe even a mild fever is normal. It's a normal response of the immune system really going into overdrive. Some people get immunizations and don't experience those symptoms. That's fine. Some people do. Some people get mild swelling and discomfort at the site of injection. So those are typical side effects of any vaccine generally. Some vaccines tend to have slightly more severe side effects. You might feel, again, some mild aches and pains for a couple of days. But again, you can also kind of make the connection that the stronger response that your immune system has in response to a vaccine, the better, because that's going to result in stronger immunity. So, and to the question, is it worth it? Absolutely, yes. Vaccine preventable diseases kill a lot of people, I'm not just talking about the COVID-19 disease. And so these vaccines, including the vaccines that are being developed and soon to be approved for the SARS-CoV2 virus, have greatly reduced the potential to get this disease. And not everyone, but some people that get COVID-19 die. We've looked at the numbers, and that's the sad part of my story, was not just the number of people who have the disease, but the mortality rate as a result. And these vaccines have the potential to greatly reduce the amount of mortality, the number of deaths as a result of this virus.
And one comment I'd like to make about this, and this is sort of the whole herd immunity and the social agreement, is one of the problems with people getting sick is how it's overwhelming our social systems. And so vaccinations helps make sure that hospital beds are available and that medical people are available to help other people who have other diseases and not just COVID. So we are overwhelming our medical systems and our public health systems because of this virus, even though the mortality rate might not be, through the roof. It still really is affecting our social structures across the world.
Okay. We're over time, but I'm going to throw one more question in there before we wrap it up. And maybe while we close closing words, maybe if you want, you can go in and continue answering questions certainly in the Q&A if you're able to hang around. Last question: if a vaccine is distributed, so kind of looking ahead, it's distributed, are lockdowns still going to be necessary, or is social distancing, wearing masks until we reach herd immunity going to be sufficient?
I'll take a stab at it, and then hopefully the other my colleagues can help. We will still need to maintain mask wearing and social distancing until herd immunity is established. Now, I think that as far as, the different tier colors we have across the state, and right now we're in like the high alarm purple, possibly as long as case numbers are going down, mortalities are going down, and hospital beds become more available, and we're not putting such a huge strain on the hospital healthcare systems, we could start to move down into the red and into the orange and into the yellow over time. And I think that having the vaccination is just going to basically speed up that process and make it happen more quickly. But yes, we will have to maintain social distancing. We will have to maintain mask wearing until we achieve that herd immunity state for our population.
Wonderful. Okay. Well, gosh, like I said, I want to thank everyone who wrote questions into our Q&A. It exploded, and we have, we could go on for 2 or 3 more hours probably answering questions. But we're gonna try to end as close to the time we said we were gonna. We want to thank our panelists again. Professor Burke, Dr. Ramey, Dr. Cude for participating today, all our background assistants and keeping us up and going with Sean and the team. And I just want to say we'll have another series of events in the spring, the Science Talk events. So look forward to that. You can keep checking our website for more information. And I'm not sure if Dr. Thule is still available if he wants to say a couple closing words. But again, thank you.
Great. Anthony, not much to add other than let's give a virtual round of applause to our faculty panelists today. They were outstanding. I know we can't see everyone's videos, but hopefully you're all clapping along with us. Looks like we've had hundreds join us today, and I'm looking at some of the names. We have a number of faculty, many students, lots of staff, some community members, local principals, school officials. So it was a very representative and diverse group today, and I think everybody benefited from the knowledge and expertise shared by our panelists. Just a shout out to Anthony too. I think Anthony did a great job both in wrangling this and in doing the Q&A. So thank you, Anthony Michaelides. And Sean and the team, as Anthony noted. For the spring, if you enjoyed this, watch our calendar, Canyons.edu/Canyon Country, and join us for chemistry talks, environmental science talks, feature with JPL, and who knows? I think COVID is certainly going to be around in the spring. Maybe we can have some of our biologists back and do an update on this in a future semester if they'd be willing. But appreciate everyone, appreciate our faculty and staff and just the college making this possible. And stay well, stay engaged, and hope you have a great holiday.